In part 1 of this mechanism of action animation series on restoring cortical network synchrony in the treatment of cognitive impairment associated with schizophrenia, or CIAS, we explored the connection of CIAS to other symptoms of schizophrenia, as well as the profound impact that CIAS can have on the well-being and outcomes of patients with schizophrenia. Here, in part 2, let's take a look at the cortical network thought to underlie cognition and how the imbalance of excitation and inhibition contributes to CIAS. Within the prefrontal cortex, or PFC, is a network of excitatory glutamatergic pyramidal neurons and inhibitory GABAergic interneurons. The synchronized firing of these cortical glutamate neurons is vital to critical cognitive processes, including working memory, sensory processing, and executive functioning. The glutamatergic pyramidal neurons receive inputs from a myriad of neurotransmitters coming from assorted neurons throughout the brain. With so many different signals coming in, how can the cortical glutamatergic pyramidal neurons possibly fire all together in synchrony? Here's where the GABAergic interneurons, and specifically the parvalbumin positive, or PV+, GABAergic interneurons in this cortical network play an essential role. The PV+ GABAergic interneurons synapse onto the glutamatergic pyramidal neurons, where they release the inhibitory neurotransmitter GABA, which acts on GABA receptors on the pyramidal glutamate neurons. But what stimulates the PV+ GABAergic interneurons to release inhibitory GABA? It is, at least in part, the excitatory glutamate coming from those very same pyramidal neurons being regulated by GABA. The excitatory glutamate acts on N-methyl D-aspartate, or NMDA, receptors found on the PV+ GABAergic interneurons. When the PV+ GABAergic interneurons hear a lot of excitatory glutamate, they react by releasing inhibitory GABA, which inhibits further firing of the glutamatergic pyramidal neurons. This GABA-driven inhibition leads to less excitatory glutamate release by the pyramidal neurons. With less excitatory glutamate acting on NMDA receptors on the PV+ GABAergic interneurons, the PV+ GABAergic interneurons are not stimulated to release inhibitory GABA. With no GABA to inhibit them, the glutamatergic pyramidal neurons in this PFC network are free to fire, and this reciprocal cycle of excitation and inhibition continues. The resulting oscillation of excitatory glutamate and inhibitory GABA, otherwise known as the excitatory-inhibitory or EI balance, results in synchronous firing of the glutamatergic pyramidal neurons in the cortex. We can actually see these oscillations on electroencephalogram, or EEG, as gamma waves. Looking at these 30 to 100 Hz gamma oscillations, you can visualize the EI balance. Here, at the peaks, is where the PFC excitatory glutamatergic pyramidal neurons are all firing together. And here, at the troughs, is where all of the PFC glutamatergic pyramidal neurons are not firing together because they are being inhibited by the GABA coming from the PV+ GABAergic interneurons in the cortical network. We see these gamma oscillations on EEG in healthy individuals during performance of working memory tasks. Not only do the glutamatergic pyramidal neurons in the PFC network form the basis for the EI balance and gamma oscillations via their excitation of PV+ GABAergic interneurons in the network, as you can see, these glutamatergic pyramidal neurons also have axonal projections to other brain areas. Their release of glutamate in other brain areas stimulates activity in various circuits, such as the mesolimbic and mesocortical pathways. Normal functioning of these neural circuits relies, in part, on the controlled release of glutamate coming from the synchronized PFC network. In the brains of patients with schizophrenia, firing of the glutamatergic pyramidal neurons in the PFC network is asynchronous. What is happening in the PFC network to cause such a disruption? Although the glutamatergic pyramidal neurons are there and firing, their regulation by inhibitory GABA is just not happening. Even though glutamate is available to stimulate the cortical PV plus GABAergic interneurons, they are not hearing it. You see, many of the NMDA receptors on PV plus GABAergic interneurons are hypothetically hypofunctional in schizophrenia. Thus, the PV plus GABAergic interneurons are not adequately excited by glutamate and therefore do not release inhibitory GABA. With no GABA to inhibit them, the pyramidal glutamate neurons fire at will, driven by the neurotransmitter inputs coming from other brain areas. As a result, the EI balance is imbalanced, and synchronous firing of the PFC pyramidal glutamate neurons is lost. We can see this loss of synchrony on EEG. When patients with schizophrenia perform a working memory task, the gamma oscillations are disrupted and correlate with CIAS. Notably, the EI imbalance in this PFC network causes unregulated glutamate release not only within the network, but also in other brain circuits, including the mesolimbic and mesocortical pathways, which are thought to be involved in positive and negative symptoms of schizophrenia. Just as CIAS highly contributes to all symptoms of schizophrenia, as we discussed in part 1 of this animation series, the hypofunctioning of NMDA receptors in the asynchronous PFC network likewise impacts all symptoms of schizophrenia. Coming up next, in part 3 of this animation series, we'll learn more about the NMDA receptor so that in part 4, we can fully appreciate the therapeutic potential of using glycine transporter inhibitors to boost the NMDA receptor coagonist, glycine, in order to restore the cortical network EI balance and ameliorate CIAS.